Hi guys, welcome to classification processes. This video is part 2. In part 1, you look to recognize the fact that biological classification can be hierarchical, okay, and based on different levels of similarity of physical features, methods of reproduction, and also molecular sequences. It's kind of like an intro. You also looked at the fact that the similarity of physical features in the Linnaean system is often used. So in this video, our goals are, firstly, we are going to describe the classification systems. This time we're going to go into greater detail for methods of reproduction, looking at asexual, sexual, K and R selection, and also the molecular sequences, so molecular phylogeny, also called cladistics. So that will be our first learning goal. The second thing will be to analyze data from molecular sequences to infer species evolutionary relatedness. Now, some of these points actually do extend over to the next unit, Unit 4, so good for you to know them and know them well. So with reproduction, many species have evolved different reproductive strategies to maximize their chances of dominating an ecosystem. So an organism's goal is not just to survive, but to be able to survive long enough to reproduce and pass on their genetic material to the next generation. So what reproductive strategies can you think of that organisms have evolved? Now, you have kept your mind out of the gutter, and one of those strategies that came to mind was asexual reproduction. So in asexual reproduction, this is where some organisms can produce offspring that are genetically identical clones of themselves. And this is the key idea here in asexual reproduction, this little bit here, that they are genetically identical. So they are clones of that original organism. And they can achieve that through a variety of ways. So first one is binary fission, which is in prokaryotes, so bacteria. In here, basically, they replicate their DNA and then they split via cytokinesis. So if you remembered your mitosis in year 11, you'll remember that this is kind of similar to this idea that the genetic material is replicated and then it's dividing by cytokinesis. However, the key difference here in prokaryotes, again going back to some year 11 knowledge, is prokaryotes do not have a true nucleus. So remember, in mitosis, it's all occurring in that nucleus where those chromosomes are replicated, divide, that nuclear envelope breaks down, they get separated. So it's not counted as mitosis in this case because prokaryotes do not have a true nucleus. If you look at the next example, you've got fission, so done in protozoa. This is very similar to binary fission. However, in this case, you can see I don't have any brackets saying no mitosis. This is basically occurring via mitosis. So the same way that our cells grow and develop and all of those things happen there, with protozoa, that's how they're building more copies of themselves. So they'll simply replicate those chromosomes, divide by cytokinesis, just like in mitosis. Another example is fragmentation. So here you've got some flatworms, and what they're doing, these flatworms, is basically they'll divide a part of themselves. And then that part that splits, this will just regenerate just like here. So let's just say here will regenerate this tail part. Here, this bit, well, it's going to regenerate this head part. So whatever part of it is divided, it'll then just regenerate to fix the missing parts. So again, that's going to be a genetically identical clone for that parent. The other one main method of asexual reproduction is budding. And this is done in, say, cnidarians, so your corals. And here you get the organism growing and developing on the original one. And this is just again sort of this growth, so via mitosis, and this will be genetically identical. Another form of asexual reproduction is parthenogenesis. So we break down that word. Here you've got parthen, which means virgin, and genesis, just like in the Bible, means creation. So this is creation from a virgin. And this is a natural form of asexual reproduction, which growth and development of those embryos occurs without fertilization via sperm. So hence virgin in this case. So how does this actually happen? Well, in the case of here, this whiptail species, they are all female. So every individual in this group of lizards is a female. So there's two ways in which we can consider parthenogenesis. And I'm going to use these two words. The first one is haploid and the second one is diploid. And these are referring to the chromosomes. Now you might remember chromosomes are the storage of DNA. In humans we have 46 chromosomes or 23 pairs of chromosomes. When you form sperm or egg, we say that that is haploid because it only contains one set of chromosomes, i.e. the 23, and in the full-grown organism like me, all right, we have 46, so it's 2 pairs of chromosomes, hence di meaning 2. So in some bees, what happens is they'll have what we call haploid parthenogenesis, which basically the males in this case just are haploid organisms. So no sperm actually fertilizes them, and the males just contain half the chromosome numbers of the females. So they're haploid there. And when a female, a bee is a female, it means that that sperm has actually fertilized that egg. So the males are all haploid. The other way this can happen when we consider diploid parthenogenesis, like these lizards, which do have 2 sets of chromosomes, during this process called meiosis, which is different to mitosis. Mitosis is basically that a cellular duplication, so you're getting those chromosomes being duplicated or replicated, and you get 2 sets of them in each organism. Here, what happens in meiosis, when you form sperm and egg, you're getting 1 set of chromosome from the original cell which it was produced from. So in diploid here, what goes on is basically, let's say I've formed a haploid oocyte, which is like an egg. It basically doubles its chromosomes, so now it has your 2 sets. So no sperm happens, it just simply doubles them there. The other way it can happen is when you're undergoing meiosis, you tend to form multiple oocytes, or like with sperm, through each stage of meiosis. So here what can happen is 2 of these oocytes fuse together, and you've got that organism. So you can see no male is required. Now, how much detail on parthenogenesis do you need to know? Well, you don't have to go into all of this. This just shows you how it can work. All you have to understand really is with asexual reproduction, you're getting genetically identical organisms. What's the benefit? Well, you do not need to spend and waste time finding a mate. You can quickly grow and develop without that, because that can be time consuming. There can also be a cost to mating, energy expenditure and all of that. The key disadvantage, though, is because these organisms are genetically identical. If there is some change or variation that occurs in that environment, they are less likely to survive. With sexual reproduction, as we'll look at soon, you get a greater diversity in organisms, so they're better able to survive when conditions change because there's more traits available there. So that's kind of the key thing. Asexual reproduction, form of reproduction that we can group organisms in when classifying them. These are where they produce genetically identical offsprings, and there are some pros and cons to that. So the next form of reproduction is sexual reproduction, and this is where organisms need genetic material from 2 parents to produce offspring. So here you're getting the female and male and exchanging that genetic material. Now, we break them into 2 main groups, and this is what part of the learning goal. So the first one, or group of organisms, they may apply this strategy. So some organisms produce very large numbers of offspring who survive without parental involvement. So basically, they produce all these offspring, parents, duck off, nice and lazy. A lot of these offspring grow, but many of them will die. So they have that high mortality rate. This is compared to K selection. All right, so in K selection, they produce few offspring who rely on their parents for an extended period of time. All right, so you might remember this. R is kind of like rude parenting, all right, because they just, duck off and leave all these offspring. And K is kind parenting, where that parent spends a lot of time and involvement with their offspring. That's one way. You could come up with your other for R and K, but they're the basic 2 differences there. So when we look at R strategists, the key idea is they produce large numbers of offspring with little parental involvement. All right. So examples include grasses, corals, insects like particularly here your locusts, rodents. All right. So by producing large amounts of offspring, what's the reason for doing this? Well, they're going to be able to, therefore, with the high mortality and predation that might occur, they'll still have some offspring that will survive to reproduce. It also means very little parental care or involvement from those actual parents, so they're using less energy. So the key idea here of our strategists, large number of offspring, little parental involvement, but many will die. So they have that high mortality rate there. Now this is different to K strategists, which they tend to live in more stable environments and produce population densities which are as high as the ecosystem will allow. When you start to look at population ecology, we'll talk about this thing called carrying capacity and the idea that there are factors, limiting factors, which can therefore reduce the amount of population or keep it at a stable level that population can grow. So these organisms, if you're a case strategist, tend to have long life expectancy. So they produce few offsprings in which the parents invest extensive care in. Think about the elephants and the care that they produce to their offspring there. So they have the ability to compete successfully for those limited resources which are available, where our strategists tend to be a bit of a boom and bust. They produce large amounts, clear out an area, and then move on. So case strategists, different, long-lived, high parental care, lower mortality rate there because they've had that parental involvement in there. So this is a table just summarizing this. I strongly recommend just putting this table into your books as a great little summary that you can refer back to. So we can see some of the differences. Short, long-lived, okay? Tend to be small in size, tend to be larger in size. Fast maturation, so that means they grow and develop really quickly. Slow maturation. Reproduce at an early age, tend to reproduce at a later age. Think humans, we tend to be later than earlier there. Produce many offspring, produce few offspring at a time, little care of offspring, extensive parental care, not strongly competitive for resources, strongly compete for the resources in that ecosystem. One of the issues here, it is very, here, two separate ideas, and that's not actually how it works, because there are some organisms which sit in the middle. So if you consider plants, for example, so trees are long-lived in some cases, so we're looking at some of these conifer trees here, long-lived, strongly competitive for resources, which are those K attributes, so those kinds, or the parenting attributes there, or however way you want to remember it, but they also produce large number of seeds that get dispersed far away from the parent plant. They don't want them anywhere near them because they're going to potentially compete for them, which is a very art strategist trait. And therefore you can see they kind of fall in the center, which is why they tend to use a continuum, right? When many species fall between pure R or pure K strategists. So think about corals here, all right? They're going to be a real high number of offspring, little parental care. And as you move along, think fish, you've got rodents, some mammals, and then the primates here, real low number of offspring, but low mortality rate, real extensive care. Hopefully that shows you the key differences there between asexual reproduction and sexual, and also R and K strategists, that you can clearly see some differences. Now moving on to our next learning goal, which is looking at molecular sequences. So all organisms on Earth are related to one another, which basically means at one point in time they had a common ancestor. So that's a species of organism whose offspring diverged over time. So there was once a common ancestor, and over time we've diverged or branched off here, and now we have this large variety of organisms that exist on Earth. Different organisms share what we call molecular homologies as well as structural ones. Okay, so homologies, think structural features when I say that look similar. Molecular, think molecular features which look similar. So these are biomolecular elements. Generally, when we're talking about this, we're looking at things like genes, which are used to test a relationship between organisms which can demonstrate common ancestry. So how closely organisms are related to one another. Now, if you have followed a bit of biology, you understand that we have DNA in our cells. Those DNA, via these two processes, which we look at Unit 4, transcription and translation, produce amino acids. So they are a biomolecular element, these amino acids which form the basic components of proteins. So if you look at here, you can see that rhesus monkeys have 95% similar amino acids to humans, which clearly show that they are similarly or more closely related compared to lamprey, which only have 14% similarity. So hopefully you're getting this idea how molecular sequences can be used. Let's go into a bit more detail. I have to give you a tiny bit of genetics so you understand it. Obviously, it will become a lot clearer in Unit 4, and we do touch on this again. One thing you can consider is a karyotype. So this of an organism is a description of the number and arrangement of DNA into chromosomes, present in a cell. So this is a human cell, this is a karyotype, which just shows our 46 chromosomes laid out there. Now, each species has a set number of chromosomes found in each cell of its body. So you can see our chromosomes as themselves can be used in some way to assess similarities, particularly when you can compare some of the chromosomes themselves between different organisms. Not a great use, and we'll see you get a bit more finer detail, but something that can be used there. Now, within each chromosome, it is made up of DNA, so a molecule which is wound around these things called histones, which we're going to look at again in more detail, and it contains the genetic code which can be translated into proteins. So basic idea, in your cell you have a nucleus. In that nucleus, it contains chromosomes. Those chromosomes contain tightly wound DNA, bits of DNA code for genes which make proteins. Hopefully that just sets you up with enough background knowledge there. So by looking at these similarities and differences between DNA sequences, scientists can actually help classify organisms. One of the examples here, these two birds, so these are the streak-eared bulbul, they were once considered the same species. And if you consider the idea of Linnaean system, you'd look at structural features and go, boom, similar. However, with DNA sequence analysis, we can understand that they are not actually similar species. And the fact that they cannot interbreed as well, which is one of those key classifications for classifying something as a different species. But DNA analysis has enabled us to now actually truly understand how different these two organisms are. So differences in DNA are actually caused by these things called mutations. So mutation is basically the changing of the structure of a gene. If you remember, DNA is made up of these different bases, A, T, C, and G, and they form the genetic code. Now, all I have to really do is change that A and that's there to a T via mutation because it's copied incorrectly, and I now have a mutated gene. That could produce an abnormal protein, potentially no protein. That abnormal protein may be beneficial, it may be good to the organism, or it may not be good to that organism and it doesn't last. However, beneficial mutations are passed on and preserved, and we have them throughout our life history record. So a conserved sequence is considered a DNA or protein sequence that is preserved across species due to optimal function. So get this down because we will be talking a lot about this idea of conserved sequences. So what does that mean? It's a sequence that doesn't really change across a whole lot of different species because it's so vital to function. So basically, when you consider some conserved sequences, if you stuff them up, the whole organism's pretty much stuffed up, so it doesn't exist. So it gets conserved because it's beneficial and it gets passed on. So when we look at this, we're looking at this particular histone, okay, and we're looking at residues 120 to 180. So what is a residue? Well, it's a single unit that makes up a polymer, so it may be a single amino acid in a protein sequence. So it might be the amino acid glutamic acid or valine or serine or any of those. So here we have these different amino acids, and we can see that this sequence is fairly well conserved. So if you look, there is a lot of similarities as you go down between these different organisms. However, there are a few differences. So the fact that there are some differences there show us that these organisms are less closely related. So a human and a chimp are closely related to one another because you go through, well, they're all the same. However, they're different compared to this mouse and cow, so you can say that they're less similar or less closely related compared to these based on this histone analysis here, on this conserved sequences. And we tend to use conserved sequences because hopefully you get the idea that they generally stay the same. So we can clearly see differences when they become apparent. So a conserved sequence, and something that's quite conserved in humans, is mitochondrial DNA. So this is DNA which is found within the mitochondria, and that tends to have a lower mutation rate than other forms of DNA found in the chromosomes within the nucleus. So they are assumed to accumulate mutations at a constant rate over time. And this becomes very beneficial because if mutations tend to occur at a fairly consistent time period, so over this many years, this many mutations occur, scientists can now track or time how long an organism might have diverged away from another one based on the differences of mitochondrial DNA or other DNA used. So it kind of provides what we call this molecular clock, so we can actually start to date different things. So hopefully you can see here molecular sequences are really good for classifying, because we can see how different an organism is. We can also see how similar they are to other organisms, and we can also track and date certain ages and events that have actually occurred. So when we consider DNA, as I said, it acts as a molecular clock, and that starts when two groups of organisms diverge or split apart. Okay, so if we consider here yeast and primates, this contains protein sequences for cytochrome C found in mitochondrial DNA, residues 5 to 25, so those amino acids there, for yeast and primates, which diverged 1.5 million years ago. So when we consider these differences here, that enables us to start to date the time that this divergence or splitting occurred between yeast and primates. Hopefully you get the idea of molecular sequences and how beneficial they are. So let's start to analyze some molecular sequences. When you look at this is showing the differences in sequence of 140 nucleotides in the same small DNA segment from 6 mammals. And it shows 100% similarity between humans and chimpanzees. You can see there's not a difference there. However, you can see that there are 6 differences between human and rat, and at least 1 difference between human and guinea pig, cow, or dog. So based on that information, you can see that humans and chimpanzees are more closely related compared to the rat, which would be the one that they're least closely related to, because there is a greater number of differences in this conserved sequence of DNA here. I am hoping this is making sense. It's quite complex and quite a difficult idea to wrap your head around. If you're getting it, you're doing well. So similar to DNA, protein can be used in the classification. So the greater number of similar proteins in that organism, once again, the closer the relationship between those organisms. So here we can consider that there's only 1 difference in the amino acid for hemoglobin between humans and gorillas, so that would be more closely related to gorillas compared to, in this case, the rhesus monkey and the mouse, chicken, and frog, which contains the greater number of differences. So I can use that particular type of molecular sequence, so in this case protein sequence, different a bit to the DNA sequences there, to show how closely related organisms are. So a protein that is well suited to function, well, it is going to be conserved, just like DNA. And again, DNA is the thing that codes for these proteins anyway, while other traits that are less vital around it may evolve or change. And this is simply the fact that good things will stay because they're good, bad things won't remain because they are not beneficial. So these advancements in DNA and protein analysis have actually enabled changes to be made in traditional classification of organisms and their phylogeny. So Linnaeus, who is considered the father of taxonomy, he made up a class called reptiles, which included, the extant amphibians, crocodiles, lizards, snakes, and turtles all together. And he based this classification on their posture. So any vertebrate that crawl on the land, dragging its tail and on the belly, he classed as a reptile. However, we soon realized that amphibians were far different from other reptiles. And other advancements came in as we started to understand DNA, started to get this idea, the fact that birds were actually closer in anatomy to crocodiles than they were to other reptiles and mammals. And this caused a change in how we classify things. So you can see here, birds are now classified here closely related to crocodiles. And birds are very closely related to the dinosaurs. So as you can see here, for example, birds are now classified as true reptiles, which is very different to how they were originally classified. And this has been based on some of the advancements that we've had in DNA and protein analysis. So let's start to look at some of these examples, see if you can apply now what you've learnt. So the analysis of molecules such as DNA and proteins can be used to determine the evolutionary relationships between organisms. Using this lot of sequences, so these LDH, looking at these residues, amino acids between 195 to 211, explain which organism is most and least closely related to organism 1. Stop the video, have a go yourself, see if you can figure it out. Now the answer, organism 3 is the most closely related to organism 1, as there is only 1 sequence different. So if you have a look up here, you can notice that the only difference is this first one, which is kind of sneaky because you start looking at this one initially thinking, yeah, it looks quite similar, but you notice that there are more differences. So here there is only one difference. So it is the one based on this molecular sequence that's more closely related. Organism 7, however, is the least closely related, as there are 13 sequences which are different, all right? And as a result, the greater the number of differences, the least closely related they are based on that particular sequence there. Having a look at one more just to drive home some of these points. Explain which animal is most closely related to a human, and explain which animals are most closely related to a frog. Pause it, have a go yourself. I'll go through the answer in a second. Looking at this, which animal is most closely related to a human? As we go through, you'll notice that the human and pig, they do not have any differences there. So based on this molecular sequence, those two are the most closely related. And most closely related to a frog, this is kind of a bit difficult as you go through noticing a few differences and similarities. But if I've checked through, it really should be the human and the pig. Based on this sequence here, using this sequence, a frog is most closely related to a human and pig there, because for the others, I think there's a few more differences. I think because you've got this one here down there with this, and here I think there's a few others. So those ones there are the ones that are the most closely related, although it may also, oh no, you've got the I, the dog's got the V. So yeah, it is human and pig, which is most closely related to the frog there. So guys, hopefully that has helped you understand the ideas of reproduction, how they can be used to classify. So we can group organisms into asexual and sexual reproduction. We can group them into R and K selected strategists, all right? The other thing that can be used to help classify is molecular sequences. And understanding here that when we look at some of these conserved sequences, so sequences that don't tend to change too much, we can see the different sequences which are similar between organisms, and they show a more closer relationship compared to those sequences which there is a larger number of differences there. That should hopefully help and get you through that little bit of content. Stay tuned for episode or part 3.